Hello, everyone. Now I will demonstrate how to use our behavior analysis system software VisuTrack. First, we open our software on the desktop. Click this icon. My presentation software is fully modular. Here are some of the experiments we have done recently. They will be displayed on the left. Up to 5 will be displayed. For some reason, some experimental files may be deleted by you and cannot be opened here. Let's create a new experimental file for demonstration. We use an offline open field video file for demonstration. The open field experiment mainly analyzes the movement trajectory of mice. This part is the pattern analysis selection, and we have to choose a based on movement trajectory for demonstration. If the test is about the forced swimming behavior of mice, then we should choose based on activity. This is the default storage path. The software will generally create an experiment folder under the release folder. This is the note information. You can fill in your experiment information. This section is for data management, which is used to manage multi-channel records. Our software supports up to 4 channels of real-time recording. If you test 4 open fields at the same time, and the marking information is consistent, you don't need to select it. The software will use the same mark to mark 4 sets of information by default. If you test 2 open fields and 2 elevated platforms at the same time, select this section, and the software will display 4 blocks for you to mark at the same time. Select the video source. Generally, there are 2 parts: real-time recording and offline video analysis. Here, we choose the second option. Open a video file that you have prepared in advance. This is an elevated platform plus cross-maze test for mice. There is a change in this video. The cross-maze moves at 24 seconds in the video. After that, the entire maze is fixed in this position. Because the maze is fixed in this position after 24 seconds, our next demonstration operation is 24 seconds after the start of this offline video. We start by dividing the area for analysis. Play the video to a fixed position in the maze, then click on the area selection on the left. There are many selection tools on this interface. We first use a rectangular selection box to select the area of the maze. As shown in the video, the vertical arm of our cross maze has walls, while the horizontal arm has no walls. After we finish using the rectangular selection box, we can define different selection boxes on the right. We define an open arm area, a closed arm area, and a cross area. Generally speaking, the cross area has no reference significance. However, in order to obtain more accurate experimental results, a separate intersection area is set. After creating the area group, select the open arm, and then select the 2 horizontal arms. The same applies to cross area and closed arms. If you think the display is too large or too small, you can click on the video source, then right-click on the video to zoom in or out. Then identify the mice. Click on the identification setting on the left, then click on the Smart Identification on the right. Drag the video progress to after 24 seconds to select the mice. When we move the mouse to the video screen will freeze. Draw a small box around the mice to identify the mice. The current recognition position is a center of gravity. We can use 3-point recognition. The head point is usually blue, and the tail point is usually green. Here is the dark threshold selection. We can choose the appropriate dark threshold according to the video display effect. The higher the threshold is adjusted, the smaller the range that the software can identify. Here is the tail filtering parameter. Because our test does not need to consider the tail, it can be directly set to 0. Here is to select the target size. The current mice size is 1481. The minimum supported is 10. For example, these mice just defecated at this location, and here is a mice droppings. But it is below 10, smaller than this blue box, so it will not be recognized by the software. Demonstrate the modification of advanced parameters. For example, because of uneven lighting, the upper closed arm of the video here is a bit dark. We can select this area and adjust its brightness. Select the Test Parameter option on the right. The first item on the left is the test duration. Generally, 10 minutes means 600 seconds. We analyze 300 seconds. This is an offline video, so select the second option. The default parameter here is to start from the start frame of the video. Our maze moves at 24 seconds, so we select the first one, starts the analysis from the current frame. The head exploration threshold is usually the default. This 5 inches means that if the mouse has exploratory behavior in the central intersection area, for example, its head, the blue dot, extends out of this area for 0.5 seconds, the software will count it as an exploratory behavior. This option is animal grouping. For example, set up a blank group of 10 mice, which is a group that is not given medication and has not been modeled. Set up a model group of 10 mice, such as mice with anxiety modeling. Then add a control group, which is 10 mice that are given medication for anxiety treatment. After creation, 30 tracks will be generated here on the right. Assume that we want to be the first mice in the blank group. Select here. If the video is recorded in real time, click the eye in the video window to select real-time recording. We are using an offline file. Click on the right to select an offline video file. Because the maze moves in the video, we click Start Analysis after 24 seconds of the video. Click Start Analysis after 24 seconds of the video. This is an offline video that can be played at 8 times the speed. Because we just selected the current frame to start analysis, we need to click Start Analysis. If we select the start frame to start analysis, the analysis will start when the video starts. We are waiting for the video to finish playing. Okay, the video has been analyzed. We can see that the mice has been wandering in the vertically closed arms. It is a relatively anxious state. It didn't go into the open area. Let's look at the data management. You can right-click on the table in the data management. There are indicators that can be displayed here. These checkboxes are the commonly used default indicators. For example, excluding the first 24 seconds, the total duration of the analyzed video is 276 seconds. There are also other parameters, such as total distance and immobility time. The time it takes to enter the open arm is also 0. Because it does not enter the open arm throughout the video, its latency is consistent with the total time of our video analysis. Right-click the table to view the data in segments. For example, if you think a certain time period is more effective, you want to view it separately. For example, you want to select the period from 120 seconds to 300 seconds. This is the data from 120 seconds to 300 seconds. Here is the data visualization. This is the trajectory parameter graph. For example, we don't want to show the outline of the paradigm. We can choose to hide its parameter. The second option is the heat map parameter table. Use color to show the mice active area. The display will default to red if the stay time exceeds 10 seconds. If you want to display it more intuitively, you can modify the parameter to display red if it exceeds 5 seconds. The third option is a behavior record sheet, which records the behaviors that occurred and the corresponding times. This is a data visualization graph. The x-axis is 60 seconds by default, and you can modify the parameters to display 300 seconds or 600 seconds. The last one is a data chart. For example, this one shows the time spent in the open arm and the central area. Other options can also choose, such as sports distance. Here we show the regional preference. We chose open arms and closed arms for comparison. The one below is open arms versus closed arms. Because the mice doesn't like open arms, the color is cold. The one above is the opposite. The color is reddish. This is a basic function demonstration. If you need to consult other functions, please contact us. We are happy to hear from you.